Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning. Today we will examine deep-sea adaptations, the suite of physiological, morphological, and behavioral traits that allow organisms to survive in extreme ocean depths. When I say deep sea, I refer to environments below the photic zone where sunlight is minimal or absent, and where pressure, cold, and food limitation define life. One primary challenge is hydrostatic pressure. At depth, pressure increases by about 1 atmosphere every 10 meters and can reach thousands of atmospheres in the Hadal zone. Organisms cope with pressure through molecular adaptations, pressure-resistant enzymes, altered protein folding pathways, and small organic molecules known as piezolytes stabilize proteins and membranes. For example, deep-dwelling snailfish in the Mariana Trench possess piezolytes that maintain enzyme activity at extreme pressure. Studies of their proteins inform us about fundamental biochemical stability. Membrane composition is another strategy. Deep-sea animals often have higher proportions of unsaturated fatty acids in cell membranes to preserve fluidity in cold, high-pressure conditions. Skeletal and body form adaptations are common. Many benthic organisms have reduced calcified structures to avoid brittle failure under pressure. Conversely, some abyssal amphipods show gigantism that may relate to oxygen and resource dynamics. Metabolic adaptations are critical. Low metabolic rates, slow growth, and energy-efficient life histories permit survival where food is scarce. These organisms maximize energy conservation and employ opportunistic feeding. Feeding strategies include scavenging, predation, and exploiting localized productivity hotspots. Hydrothermal vents support chemosynthetic primary production, where bacteria oxidize hydrogen sulfide to fix carbon. A classic case study is the giant tube worm Riftia pachyptilla found at vents. Riftia lacks a digestive tract and instead houses symbiotic chemoautotrophic bacteria that supply organic carbon. Symbiosis is a recurring adaptation in the deep sea. Mussels, clams, and other vent and seep organisms host bacteria that perform chemosynthesis, supplying nutrition in an otherwise food-poor environment. Sensory adaptations are also pronounced. In near darkness, vision can be reduced or specialized. Some species have highly sensitive eyes for bioluminescence signals. Others emphasize mechanoreception or chemoreception to detect prey and mates. Bioluminescence itself is widespread and multifaceted. Light produced by photophores can attract prey, deter predators, or serve in communication and camouflage through counter-illumination. For instance, anglerfish use a bioluminescent lure to attract prey. Many midwater organisms use counter-illumination to match downwelling light and hide their silhouette from predators below. Coloration in the deep sea differs from shallow water. Red pigmentation appears black at depth because red wavelengths are absorbed by water. Thus, red animals are effectively invisible. Reproductive strategies reflect scarcity. Many deep-sea animals have low fecundity, but invest in large yolky eggs, brooding behaviors, or long larval durations to increase offspring survival. Another interesting adaptation is pressure-tolerant nucleic acids. Genomic and transcriptomic studies show differential gene expression that supports homeostasis under extreme conditions. Research methods shaped our understanding. Deep submersibles, remotely operated vehicles or ROVs, pressure-retaining samplers, and in situ observations reveal living behavior and adaptations that were unknown from dead samples alone. Applied benefits come from studying these adaptations. Pressure-stable enzymes have potential in industrial processes, and piezolytes inform the development of novel stabilizing compounds. To summarize, deep-sea adaptations span biochemical, physiological, morphological, and behavioral realms. They illustrate evolution under extreme pressure, low energy, and perpetual darkness. Understanding these strategies not only explains how life persists in the deep, but also expands our knowledge of biochemical limits and potential biotechnological applications. That concludes the lecture. I encourage you to read the case studies on Riftia and abyssal amphipods for next week.
Listen again to part of the lecture, then answer the question.
Bioluminescence itself is widespread and multifaceted. Light produced by photophores can attract prey, deter predators, or serve in communication and camouflage through counter-illumination.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.